Peace and love, everybody. What's good? Cuts One back again. Thought I'd share a few little things that I have on my posted on my Twitter. You guys can come find. It's my pinned tweet. So here's a pretty awesome researcher. We just found the comforts of madness on Twitter. He's been finding pretty good Venter stuff. And here's one. Venter even funded how to make a non-lethal bat virus lethal with the help of CDC virologist David Wentworth and Fort Detrick's own Timothy Stockwell and Kansas State vet researcher Wenjun Ma, all with Fauci's help financially. Check that out. This is what I was talking about on the live stream last night. India charges WHO scientist Soumya Swaminathan for mass murder. The beginning of accountability? ? Look who sits on the human vaccine project. There she is. Here's another little thread. These are DARPA programs predating the garbage going on, called Drastic, Preempt, and Defuse. If you go into this, the Seeker on Twitter, he has a good thread. This is the Codex Alimentarius website, or their Twitter page. And this is the one I wanted to show right here. So I'm not going to read this whole article, but down at the bottom of this article, there's a couple paragraphs that are worth noting. Peter Daszak, president of EcoHealth Alliance and the disease ecologist, described risks of disease outbreak from areas that have stressed ecosystems as well as zoos because of living density. Essentially, where humans and animals are in conflict is where disease emerges, said Daszak. This is good news because if we're the cause, we can stop it. One example is monitoring pets. He said, the world's scariest animal from a pathogen perspective is the adorable sugar glider, a kind of flying squirrel. They're so cute. We want to keep them as pets, but they come from potential pandemic hotspots. An app called Pet Watch identifies pets that are most healthy for us and for the environment. Turning to the topic of genetics and health, George Church of Harvard University and Craig Venter of J. Craig Venter Institute described growing gene sequencing and manipulating capabilities. Why would we synthesize or make radically new genomes? Asked Church. I would argue for the safety and productivity. Helpful genes can be built in. Also, we're not going to be sequencing your gene once but many times, added Church, to monitor different microbial components. Personalgenomics.org is currently monitoring the genomes of 16,000 volunteers. this is the one with the. This is articles from 2010, by the way, so this is why I'm showing this. It says, with the recent announcement in the journal Nature of the 1000th genome sequenced, Venter noted that what we need is 10 million to truly understand human variation. We also need to study the human microbiome, which influences disease. Work by Venter and others on manipulation of DNA to create synthetic genomes will help in the study and treatment of disease. Venter quote, NIH has funded our group to make synthetic pieces of every flu virus that's ever existed, said Venter. That could speed up flu VC development from several months to several weeks or days. And that's what I'm trying to show right there. That's his company. That's Synthetic Genomes. His company's called, one of them, it's called Synthetic Genomics, where they do this. Anyways, peace and love, everybody. Much love. I hope y'all are staying blessed up and have a beautiful day.